However, we can do better than that. We can include space. So, in fact, not only can we know when the epidemic started and how quick it's growing, we can actually also find out where it started. And that is really the point of phylodynamics and phylogeography. So, this is my one-slide poster for phylodynamics. So, on the left here, you have sequences combined with the epidemiological data. So here is a tree, and the different colours represent avian, swine, human sequences. And we have a nice strong molecular clock signal. And in fact, using those ideas of effective population size I described, you can start to look at how many infections you get through time. But you can do more than that, because if you integrate your evolutionary model with your epidemic model, and also with a kind of spatial network model, you can do things like calculate the sources, routes, and speed of spread of an epidemic. You can also do things like calculate the rate of acquisition of drug resistance, and you can calculate cross-species transmission rates. So I will show you kind of how you do this. And I'm going to start by talking about locations on trees. So this is our very simple little part of a tree. And this is our sequence 1 and sequence 2 as we had before, except now we know that sequence 1 was found in location 1, sequence 2 found in location 2. And what we want to calculate is, well, okay, which location was the source of these two, for these two. Alternatively, in fact, using the exact same program and same code, we could ask, instead of locations, these could be traits, and these traits, they could be species. So it could be, so the first sequence could be from birds and the second from humans, and you could be asking, well, okay, which was it, birds or humans, that was the source? So generally, in phylodynamics, I really consider it as there's two parts to it. There's the forensic scenario, which you actually started to hear about this morning, and this is really for, you're trying to ask which individual infected which other individual. And to do that, you need very dense sampling. You obviously, if you're going to start saying, oh, well, it was definitely A that infected B, well, you better have made sure you've sampled all of the individuals that it could have possibly have been. However, there's another way you can use it as well. And you can look, instead for exactly which individual infected which individual, you can look for generalized patterns. And here, this is where you're trying to quantify the overall transmission rates between locations and between hosts. And here you don't need to have quite so dense sampling. This is more for the situation of general surveillance. Okay, so back to our which location is the source. We have a time-scaled tree, and this little branch length here, that's time t. And fundamentally, we're asking, what is the probability of being in state A after time t? Now remember, now we're thinking in population genetics, so now time is going to run backwards from the present. So this is what we know, and a time t ago, which location were we in? In general, in fact, probably what you're really trying to calculate is, given that you started in state X, so here X is just location A, what is the probability of being in state Y, some other state, after time t? And you would do this using this matrix equation. Let me show you. So here X, this is a vector, and it has, think of it as a column vector. Probability of being in location A, well, because we know this is location A, that's a 1. Probability of being in location B at this particular point, well, that's 0, but it's not in B. Then we have a transition rate matrix, which describes the probability of going from A to B or B to A and all the combinations. And notice what I've written here on the diagonal. That means 1 minus then whatever else is the sum of whatever else is on there, because in fact we're going to add, everything adds to 1. And so when you see a bigger matrix, normally you don't put these in because they're obvious from all these other ones. So you have your transition rate matrix, and you matrix multiply it by your starting off column vector for what is the first location, and then you get the probability of being still in A or turning to B according to the matrix. This here, in fact, in the practical you will be able to see these values, but these values appear here. In fact, you can see I've only done half of the problem, the other half coming this way, and you add those together to give the final probability. So in a slightly more complicated example, so now I have 4 locations, and you'll see here that I actually have many instances of the virus at each of the locations on the tree. So my goal is to infer what this matrix is. It all happens in BEAST, and you'll see how this happens. But essentially you're trying to estimate those parameters on that tree. Oops. This is that same H5N1 data I showed you before, but now at each tip I've marked from which region of the world it came from. In this case I have 5 regions: Africa, Eastern Asia, Europe, Southeastern Asia, Southern Asia, each in a different colour. And I've used BEAST, and I've inferred which region the internal nodes of the tree were in, and it's been coloured. So that's the tree output. Also, again within BEAST, and here I'm just showing you the means, I'm not showing you all those confidence intervals, but there are all confidence intervals on here. I've inferred the rate matrix of going from one place to another, and here hopefully you can see, although you should be able to see better in the practical, there's dots representing the locations of the regions, and there are lines representing the strength of the rate matrix between the two, and you can start to see that there is now a transmission pattern which you have inferred from the rate matrix. Also, just to show you, you can do it by host as well. I won't show you the rate matrix of the host, but from here you can also calculate what is the rate of going from, the red is domestic Anseriforms, ducks. What is the rate of going from domestic Anseriforms to domestic Galliforms, chickens? What is the rate of transmission from ducks to chickens? What is the rate of transmission from domestic Anseriforms to wild Anseriforms? The rate of transmission from domestic to wild species. And all of this is calculated from the tree. However, I've been describing to you discrete trait models because each thing was it was either in a discrete location, it was in one region of the world, or it was in one species. However, particularly when thinking about locations, it isn't really true that they're actually discrete populations, and particularly not in this case. This is the actual real distribution of where the samples came from in that data set. And so I kind of put them into 5 regions of the world, but I mean, you know, they're not really clustered like that. In reality, they're just much more spread out. So what can we do in that? Well, in that case, we can use a spatial diffusion model. So here, instead of discrete traits, we're going to model the spatial coordinates as continuous traits. And we're going to imagine that the viruses are going to diffuse from a point source. So in fact, what you're assuming here is that the distance of the child node is some diffusion path from the ancestral node, assuming that you've allowed a certain amount of time to elapse. So from this tree here, I've marked each of the tips, the samples, as a blue dot, and the original sample as a red dot. So here, the red dot starts in the middle, and the tips have diffused, like a Brownian motion diffusion, from that central point. And again, if I plot them time and distance from the source, you can see this kind of spreading out diffusion. So BEAST will do that kind of diffusion model as well. And the output of that is a summary tree, but each node of the summary tree is labelled with a latitude and longitude. So you can plot it on this map of the world. And so these lines now, these are the tree branches, but spread out over the world. And these are the uncertainty rings. In fact, the data is not that great, but these are the uncertainty rings showing where the virus could be. So you can see that with this kind of model, you can take your samples and infer back to the start where did this start from, particularly in an epidemic situation where you would expect this kind of spatial diffusion out from a point source.